This is Cameron Chai from azonano.com, and I'm speaking to David Faddis, who's the Director of Technical Sales for Park Systems, and he's going to tell us about their XE Bio.
Hi, the XE Bio is our most recent product, and this system is aimed at people doing work in life sciences, looking at live cells, imaging in liquid, and also people that are familiar with standard optical analysis techniques. So in essence, we've combined our AFM platform, the XE Bio stage, with an inverted optical microscope. So you have the standard optical techniques that you would be used to coming from the optical microscope, and so we have phase and DIC capabilities integrated with the system. And the AFM stage is also designed to be very user-friendly and functional for scanning in liquid. So we have a variety of liquid cells that allow the user to do experiments in liquid, look at live cells. And like all of our other XE products, this system is designed with completely decoupled XY scanner from Z scanner. So the sample will be mounted on the XY stage and scanned, rastered in the XY direction. And the probe itself for the AFM experiments are mounted on the Z stage, the Z scanner, which is easily detachable for changing probes from experiment to experiment. So the probes are mounted on the bottom of this probe hand. In this instance, we actually have a liquid probe hand on the system right now. So the system is set up ready to scan in liquid. And after changing the probe, we can easily slide the scanner back on this dovetail rail and lock it into place. The unique aspect of the XE Bio is the SICM scanning mode that we have just developed. SICM standing for Scanning Ion Conductance Microscopy. Scanning Ion Conductance Microscopy allows the user to easily and repeatably scan living systems, scan a living cell in solution. And instead of using a standard AFM probe for SICM, we used a pulled quartz pipette that is attached to an electrode that is filled with electrolytic solution, buffer solution, and the living cell, for example, would be scanned in the buffer solution as well. So a feedback mechanism is set up where a constant current is passed between the end of the quartz pulled probe and the cell that's being studied in the buffer solution. So it's a very non-destructive, easily adapted non-destructive technique for scanning a live biological system in fluid, and gives the ability to look at real-time fluctuations of these living systems. So the big benefit for our XE Bio system is this new scanning ion conductance microscopy, which ultimately is combined in with fully functional AFM capabilities, as well as a stage that is configurable with most all of the commercial inverted optical microscopes that people would already have in their labs that are doing this type of work.
All right, David, thanks very much for telling us about the Park Systems XE Bio.
Thank you.